All right, welcome to the second video in this week's set of lessons. Before he nicks off to go bask in the sun, as promised, there is my cat Trevor. Um, he generally doesn't mind being held, but I think because he's pretty keen on getting into the sun, maybe that might be why he wants to leave. So there you go, bye Trevor. What we had looked at last week is we talked about the sort of structure of DNA, which this student understanding actually sort of pretty much ticks off. So I'll do a quick recap of these key ideas here, but we have covered them. Let me know if any of it doesn't make sense though. We know that DNA is made up of two strands within our bodies and within our cells, within our bodies. And we know that it's a double helical structure, and there's these phosphates and sugars on either ends that make up what we call the backbone. And then we have these nitrogenous bases, so chemicals that contain the element nitrogen that are all slightly different. And these chemicals are called adenine, thymine, guanine, and cytosine, and they actually pair only with each other. And so adenine always bonds with thymine and vice versa, while guanine always bonds with cytosine and vice versa. And one other thing we spoke about last week was that when DNA strands connect together, those two connect together, those adenine and thymines and guanine and cytosines, and they're connected by weak hydrogen bonding. And we look at this in more depth throughout this particular part of the lesson. So complementary base pairing, that idea that the adenine and thymine, guanine and cytosine work together, is super important. And it's super important because it helps us to conserve DNA, or the base sequence of DNA. So what that means is when we create copies of DNA strands, so whenever we're creating new cells that are going to have the same DNA as the past cell, what happens is the two strands of DNA will separate, and then we start to sort of build actually the new sequence of DNA to create sort of two identical sets of DNA that both have the exact same sequence. So that conservation of DNA sort of refers to like this idea that when we replicate DNA, the sequence is preserved or remains the same. Pretty much think about it as in our recipe book, we open up a page, we want to get access to that page again. So we create a photocopy. We are not altering the sequence of the DNA, we're not changing any of the words on the page. We've then got a photocopied and an original page, both containing the same information. So, this next one is about this concept of semi-conservative replication. That is a new key idea. I would put that in your in your glossaries, because I think that it's a really important sort of term, and it's something that I've seen exam questions on, and will be one of your questions in style coming up this week as well. So chuck that in, and also there's a lot of other new terms as well that go along with this slide. So I'd highly recommend that you just keep building and building that bank of tier 3 vocabulary, because the more you have there available to you that are defined, the easier it is when you're trying to put together information later down the track. So why do we actually need to replicate DNA? It's pretty simple. Our cells will age, they won't function as well as they used to, but also we need to grow. So we might need to be able to repair damaged or destroyed cells, or we just need to be able to grow and make more cells so that we can actually thrive as a species. Without those kinds of processes occurring, what would happen is if cells, once they start to age and they don't function as effectively, and essentially we wouldn't have the lives that we do, we would not live nearly as long. All right. And there was several famous scientists that really were accredited and are now accredited with the discovery of the structure of DNA. But most notably, we talk about Watson and Crick being the ones that are credited with coming up with this idea of the double helical structure of DNA, and the organization of that. There is no pencil on this slide, so don't have to worry about taking any notes here if you don't want to. What you should understand, though, on this particular slide, while there's no pencil, is you should understand that for us to be able to replicate DNA, we need to have access to that information. And so we've talked about DNA being two strands, and we talked about this idea of hydrogen bonds being weak within the strands, within the pairing of adenine and thymine and guanine and cytosine. And so what happens is we have enzymes, which are actually created by DNA in the first place. Enzymes are this other type of chemical which help to split apart DNA so that replication can occur. And so that happens with multiple enzymes actually going in, binding to DNA, and then helping those hydrogen bonds to break so the DNA can split apart, and then we can start to build the new nucleotide sequence on each strand. It's worth thinking about for yourself why this might occur not just at one point along the DNA and then move the whole way across, but it actually occurs at multiple points when we're replicating DNA. Have a think about why that might be, why there's multiple enzymes doing it at multiple points along the DNA strand. What you can see in that image, which I think is quite cool, is you can see right at the top there, you've got the darker blue represents the original strand being broken apart and the new strands, which are the lighter blue, starting to be created. Then in the second image, you can see that the lighter blue is getting more and more created. And in the third, you can see that we've got now two double helical strands of DNA, what with an older strand and a newer strand, but both containing the exact same information. This is just a second image of the exact same thing. So whichever one of those makes more sense to you, please utilize that. You don't have to worry about the term replicon. We don't use that really, but you will have to understand this idea of parent strand and daughter strand. Just note that down probably. All right, the idea of semi-conservative replication. So semi-conservative replication, that refers to when we actually create double DNA, we have in the new sequence of DNA, the new double helical structures, we have an old strand and a new strand. So when you have a brand new cell and you look at the DNA within it, every cell will contain an older strand of DNA from the cell that it came from and a new strand of DNA that was synthesized or created that is bound to the old one. And so that picture sort of really sums it up quite well. I think you can see that darker blue original strand is going to become two brand new strands, but within the two brand new strands, you absolutely have an older strand and a newer strand because the newer strand is reading or being created from the information that is contained by the older strand of DNA. And that idea of that conservation of DNA, that's where that becomes really important. Both of those strands will contain the exact same information because the base codes are universal. And so if you look at, let me get my pencil out here, my pen. So if we look at this here, let's say that's thymine, which would mean that's adenine. That means that when we synthesize or create the complementary base, that new strand will have just the opposite. So the thymine will have a brand new adenine connected to it, and the adenine will have a brand new thymine connected to it. Because those rules are universal, basically it just ensures that when we copy DNA, we make sure that we can keep the exact same base sequence going, so then they can conduct the same functions. All right, and that's just exactly the same as what I've just said. When we look at semi-conservative replication, we have an old strand and a new strand created. All right, so I did say just earlier that you would need to put some additional, or you would need to have some additional key vocab in your vocab wall. DNA helicase. So this is a type of enzyme. If you're not familiar with what an enzyme is, don't stress at this stage. We will cover what enzymes are later, but just make sure you write that DNA helicase is a special type of enzyme. That is one of the enzymes that helps to unwind DNA and unzip it or break those hydrogen bonds so that we can replicate it. We then have another type of enzyme called DNA polymerase. And if you were in my class last year, you'll know that we can define enzymes by that sort of ASE at the end. We know that they're enzymes because they have ASE at the end of their names. One of the few things in biology that actually makes some sense. But DNA polymerase is a second type of enzyme that what it does, it essentially it gets free-floating nucleotides or bases. So it says, oh, this strand of DNA here has a sequence of AAT. And so what it does is it finds a T, builds it there, finds another T, builds it there, finds an A, and builds it there, the complementary sequence. Think about it as when you're unwinding the DNA, you're reassembling with DNA polymerase being the one that's doing that work or supporting the functionality of that work. Sort of like building Lego structures that match each other. All right, and then, like I said before, that idea of semi-conservative replication just means that whenever we replicate DNA, we will have an old strand and a new strand of DNA within the actually brand new overall strand of DNA, or both of the double strands of DNA. All right, and another image. I don't think that you can have enough images to help you sort of picture an idea. This is sort of what I was trying to do when I was just doing that there. It's probably actually better than my explanation. So feel free to chuck that one in your notes, but that is a really good image for looking at how that works. That is the last video for this week's set of slides. Highly recommend that you also watch this video as well. The more information that you can use to consolidate your knowledge, the better. All right, until next time.